Okay, I think we can start. Um, well, the brief introduction is that we are a prospective working group for the Research Data Alliance. We had applied for support some months ago, and we have been accepted. So we are going to receive facilitator support and also infrastructure and landscape analysis to help us reach some milestones. So the Research Data Alliance is a big, broad infrastructure, not only in Europe, but actually across the globe. They also have a twin in the US that is called Tigris. They work together, but they are not exactly, I would say, the same. But they work together to offer infrastructure for people who would like to advance certain domains and also produce recommendation documents. So at the end of the 18 months, we are supposed to deliver some things. I will be sharing my screen and I will try to. Okay, I think you see things here. Okay. So a little bit of a historical background for people who do not know how this working group started, and a little bit of historical information. So the first discussion started during the pandemics. There were many of us who have been working in COVID-19 related projects, and one of the meeting points was an in silico seminar that we were invited to give into an IMAG, the Multiscale Modeling and Viral Pandemics group that was coordinated by Reinhard and James, among other people. And after that, we continued to discuss, and when the pandemics were over, we decided that we had an interest in building digital twins for the human immune system. So out of many initiatives, a subgroup that was focused on the human immune system emerged. And we've decided that the human immune system was a very complicated system that had no blueprint, and it was quite a challenge for really advancing in the domain of predicting human immune response. So we wanted to produce personalized models and multi-scale models that could help us predict immune response, not only in the context of the pandemic, but also in different disease-related contexts, especially for autoimmune disease, cancer-related disease, but also pneumonia, sepsis, infectious disease, and so on and so forth. So we came together and we built a working group that would meet once a week. And in 2022, among other things, we published a roadmap. So for this roadmap, we set up some challenges for the years to come. And some of them were how to develop sufficiently powerful and detailed simulation models, acquire appropriate real-world data, and how to personalize and customize the simulation models to individual patients and for particular purposes. So in the roadmap, we asked some questions. So what are the clinical goals of a specific immune digital twin? What are the scientific and mathematical challenges that we would need to address? And what is the composition of a team to carry out the project? And of course, what are the resources needed? We understood very early in the process that this endeavor was a very big one, and it required multidisciplinary efforts and groups that would include people with different backgrounds, spanning from bioinformatics, clinics, biology, but also software engineers, modelers, data scientists, from the academia, but also from the private sector. So a year ago, we received funding from the Institut Pascal at Paris-Saclay in Paris to organize a 3 weeks workshop about building immune digital twins. So we had 3 weeks that were structured around keynotes and roundtables We had contributed talks, selected talks, invited speakers, projects, but also talks about innovation and policy. And we also had a lot of training events for people that would like to brush up their skills. So we wanted to see if we could work together in a larger scale. And one of the challenges was to minimize communication barriers and shape a common language. So in 3 weeks, we had more than 100 participants on site from 19 countries with many different backgrounds and also levels concerning the careers. We continued to discuss and we continued to keep contact after the 3 weeks, and we saw that many people were eager to contribute. So we wanted to transform this working group into a consortium and to have a more formalized existence. We wanted to create a transatlantic community, bringing together researchers from Europe, US, but also other countries. And this is when we found out about the RDA Tiger services, and we decided to submit an application for having this services and actually help us reach some milestones. So it took a lot of time and a lot of back and forth because we had to narrow down expectations and propose things that were realistic and tangible, and also try to accommodate the different expertise of our senior researchers that would participate in the working group. So all these months we have been working on our case statement and defining what we would like to deliver in the first place, so in short term, middle term, and long term, because we would like to continue to work together and coordinate our efforts to bring digital twins into the clinic and closer to the patients. So, as I said, we are now in an advanced state regarding RDA. So the RDA Tiger services will be allocated for us for 18 months. Regarding our case statement, we have now passed the community review, and we are now in the tab review. So the next steps that will probably be finalized in the coming month or in the coming weeks is the council review and the endorsement. Our presence here is a little bit preliminary in the sense that we don't have an endorsement or a case statement to share with everybody. However, if you would like to read it, if you would like to access it, we can share it with you, and you are free to provide your comments. And what we would like to do now is to start working regarding the main deliverables. So for 18 months, we will have people managing the working group and helping us reach some milestones. So we have seven milestones that we would like to reach. One of them is to create an open access library where we will curate literature relevant to the human immune system. We would like to create an open access catalog and metadata of existing models of the human immune system, and develop also a prototype by establishing a simple data structure. We also collaborate closely with biomodels, one of the biggest repositories of biomodels in systems biology, with a focus of the human immune system. So we really want to map what is out there regarding the human immune response in different contexts. We also want to have this in an open space so people can access it. We are working on a collaborative website and Git, where all the code and all the data will be accessible to the community. And we are also very keen on creating educational and training events as we are already doing. We want to have a series of virtual workshops and webinars for a general audience, including the working group members, so that people who are chairs, co-chairs, speakers that really contribute with their expertise have some visibility and also explain to the general public what is digital twin technology and how can it be implemented into healthcare. Regarding the sixth deliverable is probably a follow-up of the 3 weeks Institut Pascal workshop, something like a summer school where we could bring together people working on these deliverables, but also people outside of the working group to spend time brainstorming and setting up the next steps. At the end of the day, what we will produce as a tangible result of the RDA working group will be a recommendation document, something like a textbook for people who are interested in starting in this endeavor and integrating digital twin technology into their own projects. Together with EDIT, the ecosystem for digital twins in healthcare, Lisbeth is going to discuss about it. We also would like to work on producing some proof of concept. So we would like to have a use case that could work as an implementation example, probably focusing on information, but this is something that we would need to discuss in order to see how we can have a generic infrastructure, a smart design, something that could be adaptable to different use cases, modular and fair, and very much in accordance with what the ecosystem for digital twins in healthcare, EDIT, and the Virtual Twin Institute is advocating. So as a community, as a working group, we have already spent at least 2 years working together. So we have a Slack channel, we have published papers, we have other papers and reports that are on the making or under peer review. And we would like to open this initiative to more people. Regarding upcoming events, we will host a satellite mini symposium of half a day before the EDIT meeting in July in Amsterdam. And we will also have a more local, I would say, mini symposium on digital twins for human health on the Open Days of Bioinformatics in France that will be hosted in Toulouse this summer. So these are the more generic, I would say, information that I could share, but also the more specific regarding the deliverables. And we are looking forward to our endorsement so that the working group will be more visible in the RDA website and that people could have access to all the documents in a more, I would say, easy way. I received some messages that people tried to leave comments for the case statement they could. Sometimes the site was down in the RDA. So I think that this transition phase of the RDA website did not help a lot. So I think things will only get better from now. So for that, I'm going to stop and I will give the floor to Reinhard, who has been a co-chair in this initiative, if you would like to add some comments, and then we can proceed with the presentations of the speakers.
Thank you, Anna. I just want to make 2 comments. First of all, I want to thank Anna for establishing this. She has been really the driver of this, and she has done a terrific job. I think this RDA working group will be one of the catalysts of a community around immune digital twins. Hopefully a strong connection with Edith will be another one, but this is the sort of thing that's needed, I think, to really start building a community, and I look forward to doing that. The other comment I wanted to make is to add one thing to your description of the last year's workshop at the Institut Pascal. I think one of the important features and one of the more impressive ones, I think in addition to having 100 participants, was that we really brought together modelers, clinicians, experimentalists, people from industry, people from government, and that's a very, very difficult to do. And I think this entire digital twin or virtual human, as Lisbeth probably knows very well, is to bring this community together and you know, meld the different cultures and make us work together. So I think thanks to you and others at Pascal, there was something we accomplished.
Yes, thank you very much, Reinhard. Also, you have also been very active in organizing seminars and the Lake Nona meeting. So most of the people here are really very active and very invested in bringing digital twin technology closer to healthcare and clinical implementation. So we know that it's a lot of work, and community work is also something that is always a challenge. But we are here and we are really willing to do the extra mile. We have three participants here. I don't know if they would like to ask some questions at this point before starting the presentations. You can use the chat or you can use the Q&A at the WUBA, but you can also feel free to raise your hand in Zoom and ask your questions if something is not clear for you. Otherwise, I think we are ready to start with the presentations. Our first speaker is Lisbeth Gerritz. Lisbeth is a professor at the University of Leuven and Liège in Belgium. And she's really the catalyst of creating this European effort in building the human virtual twin. So not only the immune system that we think is a major challenge, but actually the whole virtual patient, the whole virtual twin. And she has been working diligently the past 2 years for bringing different communities together and also policy making, which is another aspect of the digital technology and the challenges that we have to overcome. So without further ado, I would like to give the floor to Lisbeth.
Yeah, I'm waiting. Thank you, Anna. I need to hide this thing so that I can find the way to switch again.
I think it's in the display settings. As
yeah, but I can't see it because I had the meeting controls.
Okay.
So now, okay, cool. Thank you. As Anna said, and Reinhard, I would like to start by talking a bit about the virtual human twin, which is a possible broader context that the immune digital twin can fit in. It is also a European Commission-driven initiative. So they came up with the concept or the request for the community to come up with a proposal on the virtual human twin, and they came up with the terminology. And the background for this is a long series of investments that they've made over time into the events at the interface between digital and health, which have led to a number of roadmaps and a number of initiatives, and also infrastructures such as high performance computing infrastructures in the EU. This has also led to the creation of a number of startup companies, and here you see the small startup companies. I'm not listing any of the big ones that obviously also have in silico medicine services. And they're focusing mostly on single levels and single organs. But if you look at what science has done over time, including work on digital twin, then you see that there are many examples of where people are integrating between different organ systems, between different length scales, and going more towards an integrated virtual human twin. So the European Commission has funded this collaboration and support action that is called EDIT, the Ecosystem for Digital Twins in Healthcare, where we have to develop the vision and a roadmap for the virtual human twin, and then also a proof of concept of the infrastructure that they will further establish later on. So as it is good practice to start defining what a digital twin is, and there is a lot of discussion on a digital twin and the work of the National Academy of Science, Engineering and Technology, where they said there is the interaction between the physical and the virtual twin in a bidirectional manner, even if the feedback from the digital to the physical can be through a human in the loop decision-making process. The virtual human twin is a new concept, so we went there with a very pragmatic definition that builds strongly on what a digital twin in healthcare is. But it goes a bit further because the virtual human twin as a concept is a systematic and ever-growing digital and quantitative representation of the actionable knowledge that we have on pathophysiology, human pathophysiology. And it entails establishing a platform that will allow pooling of the resources and assets. And these are then data sets, models, algorithms, compute power, storage networks, etc. All these things that we have already developed in Europe, elsewhere, in the scope of digital twins for healthcare. Bring this together in order to develop new digital twins in healthcare, assist people in developing their twins, and also assessing their credibility, which is crucial if we want to really take it to the bedside. And this then entails the development of a federated public infrastructure, federated because there are already a lot of initiatives ongoing, like the biomodels database, that we do not need to replace, but that we need to be able to interact with. And the collection, so this infrastructure that will allow also for the collection of these resources. And all of this, of course, requires the engagement of a collaborative ecosystem. So in short, what do we think that it can look like? Well, you can search the catalog for things that are either in the repository or in the simulation platform of the VHT or elsewhere. So that through the catalog, you can find things that already have a home, but that you might not consider looking at maybe. And this is the same for linking up with European Health Data Space. You should also be able to access digital twins that have been developed in Europe in this case, because since it's a European initiative, doesn't say that other people cannot accept it or access it and use it, but there will be a requirement for European projects going forward that if they develop twins, that they should be included in this database so that there is a more permanent home for these projects. That you can use the VHT platform to match the between, if you have a model, find the right data set. If you have a data set, find the right model. Or if you want your model to talk with a model of another scale, that you can find all these informations. That you can also run those models and that it connects you with the appropriate compute resources, and that it allows you to orchestrate all of this into a workflow that you can then share with other people, or you can also take out and further develop in a more private setting towards a standalone application. So in order to do that, a lot of work is already ongoing that can be used, but also needs to be done. And how we structure this within EDIT or in the roadmap will be around technology, integration of resources, the individual resources, the APIs, metadata schema, all of those things. There is the ELSI, the ethical, legal, social issues with access and privacy, regulatory standardization, but also the social acceptance, for instance. There is the user perspective, so who will use it, what is their value proposition, the interaction with other platforms, the access and the workflows, and then there is the sustainability element and the uptake. So how can we bring this into clinical practice? How can we bring this into practice of companies? How can we build services on top of the VHT? And what is the right legal structure to preserve or to put this initiative into? Within the EDIT project, we had to develop a proof of concept infrastructure, or especially repository. And what you see here is a very generic schematic with the bottom right talking about everything that a provider will be facing. The top right is the administration part with the identity access, the access management, the curation. And then on the left is the catalog of resources using an anatomy as a means to choose your resources that you're looking for. This needs to be connected, as you can see here, through the on the right-hand side to a simulation platform that we don't need to be developing in EDIT. So you see here a very schematic overview with the front end, the field-specific services, the middleware, and then the connection to the HPC and the infrastructure as a service. This is something for which the European Commission has written out a tender of 24 million that is currently open. So currently big consortia are being formed to actually build this simulation platform. This repository that I mentioned here will be made available with the resources of the EDIT consortium mid-June, and then also other groups like, for instance, Anna and others have already proposed additional resources will be invited to contribute there as well. So we've been doing work with the industry to talk about what is their value proposition, and roughly, of course, depending on if you talk to medtech or pharma or software, you get other wish lists and other requirements, but roughly they come down to these 7 points, saying use of the resources that are in the platform, the use of the platform as a sandbox or as a benchmark for their own developments, the use of all of the supporting work that will be done on further development of the technology, but also further development of the ethical, legal, and social issues. And then the networking aspect, so finding other partners, finding new commercial opportunities. Also the clinicians that we talk to have a wish list depending on their situation. So clinicians in training see the use of it as training on virtual patients, on testing hypotheses, on pathologies. You have the clinician practitioner, for which it really needs to be included as a standalone application in their clinical workflow to allow it really to be used in clinical practice. And then you have the researchers that want to understand how they can use it as clinical decision support systems and want to invest time in investigating comorbidities. On top of this, we have been working on sustainability pathways, so not just the market and the business modeling, but also how can we keep this? How can we keep supporting this? It's not just the European Commission that's going to be funding this forever. So how can we promote the buy-in from the EU 27 to start with, so that this becomes a permanent infrastructure in the European landscape? And then work also on policy recommendations, because Europe is now passing a lot of acts, the AI Act, the Data Act, there is the European Health Data Space. They will also have an impact on how easy or hard it will be to share some of these resources across national barriers. So these policy recommendations are also part of the roadmap that we're developing. And then finally, of course, there need to be incentive mechanisms. Why would you invest time to put something into a repository or into a simulation platform? There need to be a clear return on investment, and also that is being developed. So all of this has been put into a first draft of the roadmap that we submitted or that we published in July 31st, so last year in July. And since then it has been downloaded over 2,000 times, which I think is quite a lot. It is not a very easy document to read, but we tried to give it a bit more structure. It was the first draft. We are now currently developing this further to make sure that it is complete, and then we will make a more readable version of it. We have a full technical version and then a readable version for the entire ecosystem. So what you find in the current draft is really going from the conceptual ideas that we have at present towards the future implementation, talking about what is the state of the art, what is the vision, the technology, the LC, and then the uptake, as I already indicated. And what we are now doing is filling up some of the holes in the different sections on elements where we feel either a lot of advancements have been made and we absolutely need to include it, or even though we had already written about it, it's still slightly underdeveloped. And so we need more input to make sure that we actually do justice to the work that is already ongoing in this domain. So if this at all feels like something that you want to be part of, the road mapping at the moment is open for public writing. So there is a link here, the top one, to the first draft, and then the other one, there is a Google Maps that you will find, and also on the website of Edith, you will find a tab that says Roadmap, where you can get access to the Google Docs, and you can start working in a chapter of your choice and leave your comments. We will also do a public consultation in September of the finalized draft for final input. There is still the opportunity to contribute use cases. There is a clinical survey that we are using. It's the second one of its kind, so that we are using to get further input from the clinician side. There is a manifesto that is a 2-page version of the concept of the virtual human twin with a number of asks. And we're using this. We've had this signed already by over 80 large and small organizations, from industry to academia. And we're using, or this is really a tool that the European Commission can also use with the upcoming elections to make sure that this that as per request of the ecosystem, that this will this initiative, the VHD and its related activities, will continue to be supported by the European Commission. And then finally, Anna already mentioned it, in Amsterdam we have July 15 and 16 our final event, where we will be talking about the validation of the roadmap, the addition of some elements, and then also really go in depth on the recommendations that we want to be in there and get public consensus on that. So it's not only going to be EU focused or very technical. We have, for instance, Aldo Badano from FDA that will come and give one of the keynote lectures on regulatory science tools and how they can integrate into a structure like this. And the different sessions will really be also about the policy and about the regulatory, about the users, and then about the recommendations, as I said. So you're very welcome to join. It's a free meeting. You have to get yourself there, although we also have support for 100 people that cannot cover their own expenses to fly out there. So with this, thank you very much for your attention.
Thank you very much, Lisbeth. We have a couple of minutes for questions. Would you like to ask Lisbeth something?
So I have a question. This is Reinhard. This of course is an incredibly great and broad vision. It's quite amazing. I'm always impressed how to put all these things together. So you mentioned toward the end issues about ethics and different legal frameworks for this. In my experience, almost every time I talk to people about this topic of digital twins, especially sort of non-technical people, always concerns come up about privacy, about who is getting to use these things. And of course, simplistically speaking, a digital twin is sort of the perfect data integration tool, and that makes it incredibly dangerous, potentially. And I feel that if we want to get really broad buy-in and major investment to make this incredible project work, somebody needs to address these issues. Otherwise, I feel that we'll really be hampered in realizing this, and maybe for good reason, right? People have reason to be concerned.
Yeah, I totally agree, Reinhard, with this. I think there, though, the European perspective is very different, or the European situation is very different from the situation in the US and other places in the world, because here Europe has chosen to be very protective of privacy and to be very strict in regulating it. We had the GDPR that already made it very difficult to share data across countries. And now they realized that this was not the best way of doing it, or that they were hampering research. So they created some new legislations, which is not always the right answer. But we have the AI Act, the Health Data, or the Data Act, and the European Health Data Space that includes provisions on the secondary use of data, which is for research. There is also the primary use of data for patient-specific data. But you're right, when you make a model and you integrate the data of a patient, it can also, the in silico data that comes out does not obscure all the personal information. So if we're really working on data from a patient, there we might think about working with systems like patient data pods, where all of the personal information is stored of a patient, and then these can be connected with something like the VHD if the patient provides access. And then the VHD can act upon the data, and the data that comes out of that is actually then returned into the patient data pod. So this might be a way for personalized predictions to still respect the privacy. Of course, there are all the questions about data that sits in the hospital, that you have to containerize your model and bring it into the hospital rather than duplicate data out of the hospital. So all of these questions are not new. The VHD is not the first one to ask these, and in Europe these are things that are discussed really at the broader level. But this is not the same. It makes it very difficult. But then once it's there, it's privacy by design, which is in some other contexts it's less easy to go there or less straightforward to do that.
For
Right,
the
thank
sake of time, thank you very much, Liz, and thank you, Reinhard, for the question. For the sake of time, we need to get going. So I will ask Gary to share his screen. Liz, I don't know if you would like to share your presentation.
Sure. Yeah, no problem. I will
Okay.
send it.
Okay, that would be great. Thank you. So we are very happy to have with us Professor Gary Ahn. He's a professor and a surgeon at the University of Vermont at the Department of Surgery. And he has been very active in trying to model sepsis. So he's going to give us an overview of his efforts and also some key challenges that digital twins might provide answer to this very critical unmet need. So Gary, the floor is yours. Gary? I think the connection is frozen. Gary, I don't know if you hear us. Maybe you will need to reconnect.
Do you want me to go ahead, Anna, while Gary reconnect? Or?
I'm not sure, Jasmine, that you can share your screen. Okay, now you can. I think we can move with Jasmine and
Whichever you prefer, I'm ready. So just let me know if that helps.
just let's wait for 5 seconds, 10 seconds, and see if Gary is back. If not, we continue with Jasmine and okay. I see he's back.
Sorry, everything just crashed when I tried to go to slide mode. So let me see if I can find this. And I have the same problem Elizabeth had because the bar for the slideshow is this. Okay, can you see? You do not see that. How about that? Okay.
Yes.
All right.
Yeah.
You guys hear me? Fine. Sorry about all that. So I appreciate Elizabeth's talk about the very expansive ecosystem. This is completely at the different end where I'm focusing on a very specific disease issue and using the National Academy's formal definition because there are specific aspects of that definition that are necessary to solve the problem I'm actually interested in. And I think that this kind of melding of a very expansive view and maybe a focused view that can identify specific technical challenges that you can come up with to try and solve this particular problem. Right. So what I wanted to talk about is something called sepsis. That's kind of a term for what I would rather phrase as a acute disorder of systemic inflammation. So it's associated with multiple organ failure. Other terms for it are cytokine storm, cytokine release syndrome, etc. There is a consensus statement on the definition of sepsis, which is life-threatening organ dysfunction that caused by dysregulated host response to infection. It's a significant worldwide problem. As you can see, the World Health Organization calls this a major health crisis, and it is a very significant problem. And more severe version of this, septic shock, has a mortality of 30 and 40%, even given modern care. So the current treatment for sepsis is primarily physiologic support by giving people fluids, medications to support their blood pressure, organ function, and putting them on mechanical ventilation, and the administration of antibiotics, if available, for the affecting agent if it is caused by an infection. As I noted before, the same sort of systemic acute inflammatory response can occur to things like trauma, burns, etc. as well. All attempts, since there's a lot of basic research that's been done into the underlying cellular molecular features associated with this process, people have tried to manipulate the that underlying pathophysiology using inflammatory drugs, but none of these have been demonstrated to be robustly effective across all populations. So that is not good enough. The primary treatment of sepsis is primarily supportive treatment with antibiotics and hoping that people get better. So why is this a problem? This is a problem and to a great degree because the pathophysiology that drives sepsis are a compilation of beneficial physiologic responses that are necessary to fight infection and also recover from injury. So there's a gap between the pathophysiology and the diagnosis. We saw what the consensus statement actually is, but the pathophysiology represents a large range of cellular molecular dysfunctions. Therefore, there's a gap between the mechanisms that we know are in play and the treatment of modalities that are available. And this is a gap between basic science and clinical implementation, in some ways, the most dramatic initial representation of the translational dilemma. So this is a huge challenge. So part of this has to do with looking at sepsis from a fundamental dynamic standpoint, instead of just being descriptive in things that may be happening, trying to understand what the underlying processes are and whether or not there are any ways to think about those processes that can provide insight both into why things are so difficult to find to treat this, but also as a path towards finding solutions that will. So there are certain mathematical characterizations of this particular system. Sepsis, the population is driven by a great deal of heterogeneity and variability within both the patients who are subjected to this particular disease process, not only between individuals, but also an individual over time. There are some of these underlying fundamental physics-based principles that govern how this system works, and this issue of path non-uniqueness, meaning the fact that there are multiple cellular and molecular configurations that can lead to the same systemic phenotype. That's a huge issue. It challenges the reverse engineering problem, the inverse problem, in terms of identifying what the solution space might be, and makes it resistant to many traditional means of statistics and machine learning. And this is a problem. This is a challenge. So here's an example of what this looks like. This is a paper talking about the trajectories associated with COVID-19 in terms of biomarkers and a systemic phenotype in terms of requirement for respiratory support. This looks like every other time series data set regarding this sepsis type of problem, where if you look at the the mean values, you can identify groups across a population, but for an individual patient, individual patients don't represent a statistical distribution. They are either have it or they don't have it. And the issue is that where classical statistics is looking at group differentiation, when you're dealing with an individual patient, right, you don't know where in this entire space, if you have a particular measurement, an individual patient may be. Right. So our classical statistical methods don't have a way to distinguish, if you have a sample here, which one of these groups you happen to be in, because you could be in any of those particular groups. So there's a discrepancy between statistics and analysis for understanding, and then statistics and understanding to be able to treat an individual patient, which is actual goal of medicine. So digital twins potentially represent a path forward, because the key benefit of a digital twin is the individualization of a particular model structure to an individual person. So this is the original formal definition from Greaves in 2019. And there are some things that are inherent in this particular definition, such that digital twins are dynamic, right? You're looking at something over time, where you're thinking in terms of trajectories and not just individual states, right? And then there's this common specification that underlies the system that you're looking at that allows you to simulate the trajectory, whether you use that with a mechanistic model or some sort of chained artificial intelligence model. So this report was alluded to earlier. This is from the United States National Academies of Science, Engineering, and Medicine. And for my particular purposes, I'm going to hone to this particular definition because, again, it provides a very useful context in which the challenges that are presented in this report can be directly mapped into the problem that I'm looking at, and allows cross-pollination between domains that are talking about digital twins, right? So I think when we are talking about this larger ecosystem of virtual representations of human disease, that's great, right? And I think that though there is incredible utility in silico trials, personalized computational predictive models, and these virtual tissues, and all these sorts of things. But when you're trying to map across other domains that use the phrase digital twin, it's useful to have a common terminology. So that's how I'm going to talk about this. Right, so I'm going to focus on a couple selected points from this NASEM report that are specifically related to the application of this technology to solving sepsis, which I think is critical to solving sepsis. So the first one is fit for purpose. This is emphasized over and over in that particular report, saying that the particular approach that you're going to use to making a digital twin or immune digital twin is defined by the problem you're trying to solve. This issue with respect to verification and validation, uncertainty quantification essentially means, do you trust this particular object? Right, this is essential in terms of moving forward to any sort of regulatory assessment and implementation. There are specific problems related to being able to do that for individualized models because those population statistics and predictions can't apply, right, in that particular context. The NASEM report also talks about controls and ostensible reason for DTs because the point of medicine is to treat people, not just watch people. And because of the ongoing data link that's part of the NASEM report, the idea is that these sensor data stream technologies may not exist, and therefore the creation of a digital twin system can drive that particular development forward. And this ethics data privacy issue was raised. I'm going to address that a little bit because in this particular context, I don't think it's as big a deal. So I'm not going to talk about what we actually did because in the interest of time, I'm not going to talk specifically about this implementation. I'm going to point you to this recent preprint that we have that talks about all of that, which is essentially the design specification of a critical illness digital twin to cure sepsis, right? Focused on the things that I'm talking about, right? And these are how this particular schema addresses those particular NASEM focus points I just talked about. So the fit for purpose is to identify and optimize novel control of sepsis that we don't currently have. And it utilizes a combination of a multi-scale mechanistic model of the cellular molecular processes underlying acute inflammation with an ongoing sense actuate control process, which is the data link and the control component. We have a novel method towards validation, uncertainty quantification related to biological systems. It incorporates the inherent uncertainties both in your computational representation and the data structures that you're dealing with that are ostensibly your ground truth, right? You can read about that if you want to read about this paper. The aspect of control, since it is there's heterogeneity in the patient population. Part of the reason these clinical trials have failed is because there's insufficient substratification of this group to a point where each individual patient is their own particular patient. We adopt a control policy using deep reinforcement learning. You can read about that if you wish. And this other aspect of the data link integration in terms of the continuous control problem. The idea here is that you are, because of the heterogeneity and stochasticity in the system, you're always thinking in terms of a rolling forecast cone. That cone needs to be updated with sensors, just like you would for weather prediction, and also controls that are based on what can actually work. So that's that. In this particular concept of a digital twin, since we're looking at individuals as an aggregation of measurements, right? And the way that this system works is very much offline, right? Where the implementation has to do with ensembles that fit particular personalized forecasts. The data privacy equity issues don't, or the data privacy issues don't affect this as much, because many of the features that people worry about with respect to privacy, i.e. demographic, ethnographic, sociographic types of data things, don't actually apply to this because the context of its use, right? This is where I think specifying your fit for use can allow you to address these things on an individual basis. Right, this goes to the non-uniqueness aspect. So this is a picture of what this concept would look like. You would want to test this sort of thing in the real world before you actually implement it in a particular patient. But this is kind of what the overall idea might look like. So with that, I will finish and thank my colleague Chase Cockrell and our funding agencies for this. Thank you.
Thank you very much, Gary, for a very informative presentation about sepsis and all the challenges. Do you have a question for Gary? Okay, for the sake of time, I will ask Jasmine to start sharing her screen. We can take some time to think about questions that we can ask at the end. I think it was a very dense presentation, Gary. A lot of food for thought and not very easy to really put the finger on the critical aspects to ask easy questions. So I don't think there are any easy questions for this topic. So I would like to thank Jasmine Fisher, Professor of Computational Biology at the UCL at the Cancer Institute, the University College London, who has been pioneering in executable biology since many years, building models for cancer. And she's here to give us her view on digital twins for cancer and how far are we from a clinical implementation. So Jasmine, the floor is yours.
Thank you very much, Anna. Thanks. So I'll follow the trajectory that Gary started with, talking on a specific vertical of these digital twin. Gary talked about sepsis, I'll talk about cancer, but of course, as Anna mentioned, this executable modeling approach is applicable to many different biological processes, diseases, and it's not restricted to cancer. So whatever you see here, we can also apply to other diseases. And so the focus, second, there you go. So our vision is basically to use these computer-based models or digital twins to use them to personalize treatment paths for patients, or as was said earlier, for these clinical decision support tools. And we envision this as a growing library of different types of digital twin for each type of cancer that can then be used by the clinicians. But the main thing is that they are personalized to an individual patient by their own genetic makeup. And that's how we initialize these models. And I'll use the few minutes that I have just to give you some examples of how we build these kinds of models and how they've been successful so far. So, and the focus within my lab is to study how these genomic changes drive tumor evolution under therapy. And for that, we're using these computer programs or executable programs. And these are data-driven mechanistic models that are completely transparent and completely interpretable in terms of the inner molecular making of the cells, cell signaling within the tumor and the microenvironment and the crosstalk between them. And within the lab, we are looking at tumor evolution, tumor treatment optimization, and specifically studying resistance mechanisms. And we have this growing library of different types of cancers: melanoma, a triple negative breast cancer, non-small cell lung cancer. We studied several leukemias in the past, and now we're also starting to work on glioblastoma. And so this executable biology approach is basically an iterative cycle between collecting data in the lab and integrating that into some form that the computer can read and interact with in the form of some formal specification. And on the other hand, we have these mechanistic understanding that explain the process that we are studying, and this is turned into a dynamic executable program that represent the mechanism and the working hypothesis, and then using means of some method in computer science that is called formal verification, we compare the two, but in an automated manner, and we check that our working hypothesis or a particular molecular mechanism in the first instance is consistent with the actual behaviors that we observe in the lab. And of course, the usual process is that initially the model fails. And as I always say to my students, these failures are actually good because it means that something in our understanding is missing. And then we iteratively fill in those gaps, but at least now we know what we are looking for. And by filling those gaps and then running the program again, and now when it's actually satisfying all the known behaviors, we can then design a particular experiment that can validate that prediction. So in the context of cancer, these programs, and our programs are focused on the cell signaling. We think of it as a graph of nodes and edges, and the nodes represent proteins and genes, and the edges represent the interaction between them. So we have such programs that represent the normal tissue, and we have such programs that represent the tumor cells by activating specific genes with some driver mutation and setting those nodes into a particular level. But it's important to note that we also have these outputs that the program has, and these are in the form of some cell behavior or some cell phenotype, like proliferation and cell death, and these also have a certain level. And then we can also simulate the effect of a drug. But we always do this both on the healthy tissue, on the normal tissue, and on the tumor cells, so that when we look for new targets for combination to treat or to resensitize cells to radiation, for example, we always make sure that we don't resensitize, we specifically sensitize the tumor cells, but not the healthy cells, in order to minimize the toxicity and maximize the therapeutic window. So this is, as I said, an iterative cycle. We start with the experimental data. We suggest a mechanistic model that is turned into a computational model, and we compare them. And until the models satisfy all the known data, we continue to refine it. And then in that process, we come up with new predictions that are then validated experimentally. But it's important to understand that we use two different kinds of data set to build and to test these models. So the data that is used to build is, for example, literature data or any publicly available data that talks about point-to-point communication within those networks. And perturbation data, which is a different type of data where we perturb MYC or we perturb RAS, and then we measure the level of proliferation or level of apoptosis. That's the kind of data that we then use to test these models. And only when the models satisfy all the known perturbation data, that's the point where we can start to do in silico experimentation and look for to do in silico screens for new combinations, monotherapy combination, and sequential therapies. And this is just to show you that there's a lot of emphasis on making all of these programs visual and graphical so that they can be used hands-on by biologists or clinicians, and that's what you can see here. One of such tools is called the Biomodel Analyzer, BMA, and it's publicly available and everyone can access it. It's an open source and it's a community effort. And you can basically draw these networks and then press a button and the whole execution is running at the back end, and then the result is portrayed back in terms of how things change over time and whether the system reaches a certain homeostatic stable fixed point and what does it mean biologically and so forth. And so we've been using this approach, and you are familiar with the concept of these gene regulatory networks and how the downstream nodes are actually connected to some cell behaviors. So we define these models in the form of this discrete graph, but there are also these mathematical rules, target functions that are driving the nodes up or down depending on some mathematical logic. This can be a form of an AND or an or something more complicated, which basically describe the biological rule. And in the context of leukemia, for example, you can see here that we have a certain signature that talks about a disease state, but then if we perturb in certain nodes, we can then mimic how we can reduce the disease. And so a later work was looking specifically on acute myeloid leukemia, and we managed to build a model that produces different cell lines that are either sensitive to particular drugs or resistant to particular drugs, and then use that in order to identify ways to overcome that resistance, and everything is obviously validated experimentally. But you can use these models also to look more clinically on how you can understand the tumor heterogeneity, and by understanding it, finding ways to herd it into a way that we can actually fight it better. And this is a study that we've done in the context of breast cancer, where we look specifically on the MYC, which is an oncogene, on the MYC heterogeneity, and we took advantage of the fact that you have these mice that you can turn MYC on and off. And we also build a model of the network that involves the different signalings that are talking with MYC and how they are affecting the proliferation and the death of these tumor cells. And then what happened is that in the first instance, when you take the data and you build such a model and you check that the first version of the model is actually consistent with what we see in those mice, you already see that things don't match perfectly. And one of the things that we found is that there's a need for an explicit mutuality between the high expressing MYC cells and the low expressing MYC cells. They are dependent on each other. And this is something that we didn't know, so this was validated experimentally. But then you can also, in a specific way, you can initialize the model to look at the specific subclones, the different types of cells that you have within the tumor mix. And you can look on the effect of particular perturbations with drugs, for example, on each one of these clones, and see how you can maximize the beneficial effect in killing those tumors. So in those heat maps here, what you can see, they represent either proliferation or cell death. And we look on those with respect to either normal cells or cells that are having a high MYC or low MYC or a mixture of the two. And then these are the different perturbations and how they affect the proliferation in the cell death. And then so when you do this in a monotherapy setting, you can see that you can't kill all the different subpopulations of the cells. So then you want to do that in a way that you can combine different drugs, and this is the direction of doing a drug combination. And then what you can do here is that once you initialize the model with particular perturbation with one drug, you can then start adding a second drug, and you see when you're starting to have these synergistic effect, which means that you are enhancing on the effect beyond what each drug does alone. And what you see in this more complicated heat map is basically that difference between the two, and how you can identify specific pharmacological vulnerabilities to particular combinations that will actually benefit and kill the tumor much better. And one of these combinations that we identified was a combination of MEK inhibitor and COX-2 inhibitor, and they were validated in vivo. So these are some examples. I just, because of the focus on this session on more immune digital twin, just to show you that we can also include the immune system. And this is a more recent work where we looked at melanoma and the dialogue, the specific dialogue between the tumor cells and the immune cells, and how this immune surveillance is actually going undetected, and these tumors are allowed to evolve. So we build a model that actually looks at the crosstalk between the Langerhans cells in the screen and the melanocytes, and we've identified, I'm summarizing a very long story here, but I've always put the references at the bottom so you can look up the papers, that there's this feedback loop between the two cells, and once you break that feedback loop, that's how you can have these cells to be undetected, so to speak. But then once you know that there is a need for this feedback loop, and we tested different hypotheses, and then we homed on that one, we can look to identify specific combinations that can maintain this immune response. And so we did something similar to what you've seen earlier, and we checked specific combinations of treatment and how we can then bring back the response and the activity of the Langerhans cells, and identify combinations that are specifically targeted to melanocytes, but without limiting the Langerhans cells migration. And oops, sorry. So Anna asked us to also suggest some challenges going forward, and of course there are quite a lot more recent examples that are happening within the lab, but I don't have time to talk about all of them. So I'm just gonna wrap up with some of my thoughts on what are the main challenges in this direction of these medical digital twins. And I think one of the things to remember is that we really need these high resolution kind of models at the base. And these models have to basically cater for a multi-scale level, because when we're talking about the patient, it's not just one layer, and multi-scale in terms of time and in terms of space. And this is quite a challenge. How do you bridge all of these different levels into one model? Now, when we're talking about these kind of multi-scale models, we're talking about usually hybrid models that have both discrete and continuous part, and the analysis of those is not straightforward. There are no specific formal methods that allow you to do that in a scalable manner. So at most you can do simulation, and this is another challenge. There are, of course, challenges when you want to validate, to do the validation studies for these models and their predictions. And we are finding this a major challenge on our day-to-day, and always having to come up with new methods to do the validation, because the model can actually have a lens and a resolution that we don't necessarily have in the lab, and we have to come up with very creative way to actually validate some of the predictions.
Jasmine, I would need to ask you to wrap up for the sake of
I'm wrapping
time.
up. This is my last slide. Yeah. And in terms of adaptability, this is another challenge, because you want to make sure that these models are always up to date with the latest finding. There is something that was mentioned earlier, the patient heterogeneity is something that needs to be taken into account. And the fact that we are initializing these models with the genetic makeup of the patient is great, but there are lots of other factors that need to take into account. And of course, the fact that in order for them to be used in the clinic, they need to be interpretable in order to create the trust that is needed from clinicians. And of course, that they have to have a reasonable prediction accuracy. So I will stop here. Of course, none of this work wouldn't have happened without a lot of collaborations and funders. And this is my lab. And maybe I'll leave this slide as the one that we can use for discussion. Thank you.
Thank you very much, Jasmine. For the sake of time and to make sure that James has enough time to present, we will move with James' presentation, and we will keep the questions for the end if we still have some time to have at least one question that you can all answer to. So James, okay. So we are very happy also to have with us James, who is also a co-chair in the working group. James is a professor in the Institute of Biocomplexity at Indiana University in Bloomington, USA. And today he's going to give us an overview about the challenges in multicellular virtual tissues and immune digital twins. So James, thank you very much for being here, and the floor is yours.
Thank you. I wasn't exactly sure what to say, and I tried to respond to what you asked for, which may not have been the best choice, so we'll see. So when we talk about modeling, the world that I come from is multicellular systems biology. In particular, we want to recognize that the environment that cells find themselves in, they create and respond to, and that means that you can't understand cells by looking inside of them by themselves. And we have to deal with the fact that there are a lot of single cell behaviors and cell interactions, and that in fact you cannot predict what those are from the molecular concentrations inside the cell. Pace, what Jasmine was just saying. You can try, sometimes it works, sometimes it doesn't. That's a big challenge. Makes modeling very hard because the network models are hard to connect to cell behaviors. We have a lot of physical constraints and complexity in the intracellular architecture and the environment. And these together lead to the complexity of biology. Obviously, if we want to model this, it's expensive. We typically, in our virtual tissue world, start from cell behaviors, try to go down and do the kind of control that Jasmine was talking about, try to go up and model the environment that cells find themselves in. This is the world that I live in. It is not obvious to me that digital twins, at least what I'd call online digital twins, real-time digital twins, are going to be using virtual tissues early on because they're so expensive computationally. On the other hand, virtual tissue approaches, these multi-scale agent-based approaches, are certainly going to be critical to generating biological understanding, as Jasmine pointed out, and Gary pointed out, for calibrating the simplified IDTs, for developing potentially data resources for machine learning training and so on. My world, and we were asked to talk about what we're doing personally. I build software tools. I'm involved with software tool building. We do some modeling that's of IDT relevance, and we are trying to develop tools to support our community, to support the virtual tissue side of things. And one of the things I would like to talk about is how to make sure that those things complement each other rather than overlap each other. I think we can learn a lot from what you and Anna and Lisbeth have already done, but I think there's a lot more to learn and share. So as people know, I build a software environment called CompuCell that's designed for building these kinds of models. One of the things that it does, at least on a computational level, is integrate network models of various kinds, MABUS, Boolean network models, OD models, Gillespie models, and so on, through a variety of standards. The development of TissueForge, which is now at University of Florida with Reinhardt, and was started here at IU, TJ Sego. And this is trying to build high performance modeling capability to make this kind of modeling a little bit more accessible in a practical time scale. In terms of specific models that we do related to immune digital twins, like many people back in 2020 through 2023, we did a fair amount of modeling of infection and immune response. This was a fairly detailed model of influenza response in the lungs. It was designed to be modular and extensible, so that those are components that could potentially be used in further modeling. These days, I'm mostly involved in modeling corneal homeostasis and damage. That doesn't seem initially to be a particularly immune-related project, and the simulations I'm showing here don't include immune response. But in fact, fibrosis and chronic inflammation are critical aspects of this. Failure to heal are critical aspects of this. And so actually, we're now beginning to tool up with our experimental partners and also computationally to try to build models that include immune response in some detail in this context. In this case, it's not that well studied. The cornea is a relatively understudied organ system. And so there's a lot we need to learn from better studied organ systems like the skin and other epithelia. Obviously, we all want to see these kinds of knowledge development workflows. We'd like to see virtual tissues as part of that. Certainly, these are things that we would like to do. We've tried to make our software tools available and more accessible to integrate into these kinds of workflows. Initially, they were designed to be more freestanding, and that's something we've worked on a lot. If we're going to really build these kinds of immune digital twins, we need to be able to build these models, as we've heard, reliably and reproducibly. I tend to be more on the software side of things, thinking about intercompatibility, reusability, componentization. VT world is particularly fragmented. The network world is much more unified and much more integrated. And that fragmentation makes building these kinds of workflows much more difficult, makes validation much more difficult, makes extension much more difficult. And so there are many areas where we could try to do harmonization of our approaches. Some of these were discussed by Jasmine and in the Edith proposal and others. Some of them more specific to the VT questions. There was a nice set of challenges identified by Fletcher and Osborne, which will look very familiar to people from 2021. It also shows some of the fragmentation of the landscape of tools in this world. In fact, in the past few weeks, when I did a survey, there are more than 30 incompatible tools for virtual tissues. Not all of them are used by a lot of people, but it's a bit of a disaster that way. We have a small amount of funding from the National Science Foundation called OpenVT, which is trying to develop this ecosystem. It's not exactly the same. I don't think it really is a competitor to EDITH or to these other ecosystem projects because it's focusing on this kind of, specifically on this kind of multi-scale modeling, multicellular modeling, which, as I say, I think at the moment is more on offline digital twins and scientific discovery. But I would like very much to talk to people about how we can make sure that this effort complements and supports the efforts that Lisbeth and Anna and others are undertaking. Currently, we are doing some rather simple things, perhaps. We're defining some reference models to implement across VT frameworks. We've had 2 meetings on that so far. Tomorrow, we'll be scheduling and planning a meeting to actually describe model framework capabilities. That sounds very dry and technical, but it's actually pretty important. We're working a little bit on shareable input and output formats for these models. We would love to eventually have a portable model specification language. That's not going to happen anytime soon. We'd like to have standards for metadata annotation so you actually know what is in a model and how to describe it. There are a lot of challenges which are more broadly IDT. Many of these things we've heard of from the previous speakers. Something that comes up specifically in the case of virtual tissues is that your output are movies, they're images and movies. And it's much easier to compare the level of a chemical over time with the level predicted, because you can do a least squares regression in that. It's not so obvious how you compare 2 images to each other. And so they're actually, Jasmine had a nice example where she had a box that said compare experimental result to computational result. It's not so easy to do that in spatial biology. And so I think that that's a topic of some need at this point. The other things that we've talked are general things that we've heard about before. We teach our annual course on how to do this, if anybody's interested. But really, I'm here to listen and learn from this community and see how I can help support it. So thank you.
Thank you very much, James. I would like to thank all the speakers because they really respected the time slot, and even though we started later, we really managed to have nice discussions and nice presentations. So I would like to ask all of you one or two questions, and then we can close this session with that, with your answers. We have 18 months in front of us with the support of the Research Data Alliance to focus on the human immune system and do something. So the repository and the metadata are low-hanging fruits. There are things that can be, I won't say easily done, because volunteering work is not it's not easy. I would say crowdsourcing and finding people to do the dirty work. It's a lot of work, the metadata and the curation. Still, we are determined to do that in order to map what is out there. However, you all come with your expertise and your high-level knowledge and skills in your domains. So one question is, how easy do you think it would be to implement the human immune response to your specific projects, for example, the cornea or osteoarthritis or cancer-related or sepsis-related? And is it tangible? Is it realistic to propose a generic IDT implementation that can be adaptable? Do you think that a generic immune digital twin has a sense, has a meaning, or is it a lost cause because the human disease is so complicated and so different that would require so much adaptation and implementation that at the end nothing generic would actually work? So I see Gary wants to attack this question first. Yes, Gary, please.
Well, so I think, I mean, ideally you would eventually move towards that, but I think this is why the NASEM report's useful as a guide for how you implement things, because there are so many different purposes for why one might construct a digital twin, and each one of those would focus on different scales of resolution, different components that are represented, et cetera. So I think it would be useful to have like an aggregate knowledge graph about what we know about immunity and inflammation partitioned in multiple different ways, of which someone could pull from. But I think for specific immune digital twin implementations, because there's such a breadth of application, it's hard to think of one standalone executable that would be used.
Thank you, Gary. Who would like to follow?
I don't know. I can't find Lionheart has his hand up. I was trying to find the raise hand and I couldn't find the button.
It's okay, James. You can.
So I think that having a systemic IDT that was not a spatial model, but more a disease map style of the kind, for example, Jasmine and you and Lee have worked on so much, could be very useful. But I'm not sure that the fit to purpose question is going to be answerable so easily by that. I do think that in these specific contexts, if you're doing cancer cell immune evasion versus damage to the lungs during sepsis versus the very specific immune response in the eye, where you have just the way there's a blood-brain barrier, there are blood eye barriers. Where you have very specific kinds of immune response, the resident and circulating cells are different. I think in those cases, you're going to have to try to build specific to-purpose models. On the other hand, having general models of fibroblasts of the various immune cell types that could then be customized, and understanding the basic communication strategies that cells use would be very general. And so I think the underlying scientific underpinnings and some of the components were reusable, even if the full model will not be.
Thank you, James. I see Reinhard is raising his hand. Reinhard?
Just a brief comment. I think that we could look at weather forecasting, right? So basic rules of atmospheric physics are the same everywhere, and every weather model has a core model of atmospheric physics. And that's how I think of the immune system. It will need to be customized for any particular application because the immune response is very tissue specific. But that doesn't mean that you need to start from first principles every time.
Thank you, Reinhard. Please, Jasmine?
I think definitely the answer to your question, Anna, that it is useful tool to have, and I strongly believe that by building these different modules that can then be integrated into more specific personalized models, there's a huge value. And I think this is probably the way forward, because we are drowning in so much information and trying to integrate it in so many different ways. But actually, at the end of the day, you want to do something that benefit patients. So I think this is definitely the way to go.
To end this, maybe Anna, in the roadmap we chose a very probably a misnomer, but we made a distinction between a generic digital twin, a population digital twin, and a subject-specific digital twin. And I agree that, well, you know, the word twin implies that it's subject-specific, but oftentimes these things develop through first a more generic twin, where you're happy to be aiming, especially if you develop it from a more mechanistic perspective, not a data-driven one, where you aim for your quantity that you're modeling to hit the relevant range, right? And then with the population, you try to mimic some specific quantity of that population, usually the median or the mean. And then when you go to a subject specific, it needs to hit that data. But oftentimes this is a process of further refining every time and including more data every time. If you develop a data-driven twin, this does not really always apply, and then you sometimes turn it around. You start from a patient-specific one and then go to a population one. But okay, so we just to have this aspect of there can be different levels of personalization at different stages of the development. That's why we had these concepts there.
Thank you very much for this comment, Lisa. I do believe that this working group will set the groundwork for a generic human immune response that hopefully could be adapted to specific use cases, but at least have a core of how the system responds in a set of different circumstances that could be categorized into 4 or 5 different scenarios, for example, and try to model that and provide something that can save time when you are trying to contextualize it and make it more specific. Of course, it's not going to be perfect, but if it is mechanistic, then it stands a chance to kind of stay there and be easily extended and adapted. I see Gary wants to have a comment. Yes, Gary. Gary, you're muted.
So this is a bomb throwing comment, which is that we become very enamored with our particular mechanistic models, and we have to accept that they are all the byproduct of a certain amount of bias and selection, right? And that the focus should not necessarily, particularly if we're thinking about digital twins that eventually will be implemented in the real world, this aspect of validation and trust establishment for these particular representations, which are admittedly incomplete, is something that we really need to think about strongly. And I think that set of capabilities actually is more scalable than any particular representation that one might come up with. So I think I mean, we all make models. We love the models that we make. We believe that they work. But we have to acknowledge that there are inherent biases associated with that, particularly if we are trying to do control, right? Develop therapies on those systems.
I
So
think this is a very nice comment. I don't know if we have questions from the courageous participants that stayed with us until the end. I don't know if Anu or Ramiro would like to ask a question before we close the session. Ramiro?
Thank you so much. No, I don't have any to add. Just I joined the meeting, just very curious on the topic, and it's really, really helpful. Thank you so much.
Thank you. So we are going to close the session with an open invitation to the RDA members. We would be very happy to have people joining us in these challenges, and I would like to thank the speakers and also Athena and Rebecca, who stayed with us and kept notes from the meeting. And as we said, this meeting is recorded and will play in repeat for the 500 participants of the RDA plenaries. So thank you very much. I don't know if you would like to have a final comment before closing the session.
Just thanking you for organizing it, Anna, and the community.
Thank you. Reinhard, would you like to have the final comment?
No, I think this was great. I had a question for Gary, actually, a very quick question that allows for a very quick answer. It's a sepsis version of a question I always get, and I don't like it, but it's an important question. And Gary, since you have such a well-defined digital twin project, my question is, what's it going to cost, and how long is it going to take to build a usable sepsis digital twin?
I would say $25 million and 5 to 8 years, depending on the experimental models that are available, which is what I'm working on right now.
Great. Thank you.
Thank you. Thank you, everyone. And have a nice afternoon, morning. Yes.
Thanks, Athena.
Thanks, Anna.
Bye, everyone.
Nice seeing you all.
Bye.
Thank you.